Welcome back to the deep dive. Today, we're plunging into a really fascinating paper on cell tracking. It really shifts how we might think about the very start of cancer.
It does. We talk a lot about tumor heterogeneity, how varied cancer cells are.
Right, that cell-to-cell variation.
Exactly. But this paper digs into how that happens, generation by generation.
Hmm.
And the core idea is kind of counterintuitive. It's just DNA damage isn't shared equally when a cell divides.
So, not an equal split of problems.
No. It seems to create this, an asymmetric heritable scar. It means the two daughter cells start off fundamentally different right away.
Okay, that sets the stage for heterogeneity immediately then. So the problem is, looking back at a tumor, you see the variation but not the origin story.
Precisely. Retrospective genomics gives you the final picture, the family portrait with all the different members, but you miss the dynamics. You don't see how that diversity actually arose.
And that's the gap this research tries to fill.
Yeah. They developed this method called multi-generational single-cell tracking. It pairs live imaging with quantitative immunofluorescence. Live plus QIBC, they call it.
So they can watch it unfold in real time.
Basically, yeah. They needed to see cause and effect. They engineered cells, specifically U2OS cells, using CRISPR to tag two key proteins.
Okay, what were they tracking?
They tagged PCNA, that's a replication factor. It lights up when the cell's actively copying DNA, during the S phase.
Got it. Marks DNA synthesis.
And the other was 53BP1. You can think of that as a flag the cell raises for specific types of DNA lesions, particularly ones that get passed down through division, inherited damage.
So with those tags, they could watch both replication and damage inheritance.
That was the goal. And the first thing, looking at their Figure 1 data, was to check, can we actually track this inherited damage reliably?
And could they?
Yes. The live imaging was key. It showed that these 53BP1 damage flags didn't just pop up randomly. They appeared consistently after the mother cell divided, showing up in the daughter cell's G1 phase.
G1, that's the prep phase before DNA copying starts again, right?
Exactly. And crucially, those damage markers tended to clear up just as the daughter cell entered S phase, suggesting it had to deal with that inherited damage right then and there. It's proof, basically, that inherited damage creates an immediate G1 checkpoint or crisis.
Okay, so the damage is passed on. It shows up early in the daughter's life. But this brings us to the asymmetry part. Why wouldn't that damage just get, split evenly between the two daughter cells?
Right. That's what you might intuitively expect. Symmetrical problems.
Yeah, equal inheritance.
But that's not what they saw, especially when they induced stress. This is Figure 2. They use things like an ATR inhibitor or acetylcholine to stress the cells during replication.
And what happened to the sister cells then?
Big increase in heterogeneity. The two sisters, born from the same stressed mother cell, immediately started behaving differently.
How so?
They saw asymmetric S-phase duration. One sister took longer to copy its DNA than the other. They saw unequal amounts of damage markers, like 53BP1 foci and another marker, gamma H2AX. Even downstream effects like P21 induction were uneven.
Wow. So one stressful event in the parent cell leads to two daughters that are immediately on different tracks, one potentially more damaged or slower growing than the other.
Exactly. That single transient hit creates a persistent genomic scar that affects the next generation's fate right from the start. It's a powerful mechanism for generating diversity quickly.
That really does change the picture from simple inheritance. Okay, what else did they find?
Well, they also looked at polyploidization, whole genome duplication. It's common in aggressive cancers.
Where cells end up with extra sets of chromosomes.
Right. And using their tracking, particularly the PCNA patterns, they saw that oncogenic triggers like overexpressing HRAS and cyclin E1 could drive this genome doubling through two distinct routes. This is Figure 4.
Two different paths to the same outcome. Does the path matter?
It absolutely matters. They identified endoreplication, where the cell duplicates its genome but skips the division part. Sometimes that's almost like a programmed event.
Yeah.
And then there's re-replication, which is more chaotic. The cell tries to copy its DNA again when it shouldn't, like during S phase. And that route, it's far more damaging.
How much more damaging?
Significantly. Cells that went down the re-replication path consistently showed much higher levels of damage markers. Gamma H2AX, p53 activation, p21, way more than the cells that used endoreplication. So the way a cell becomes polyploid has huge consequences for its genome stability.
So it's not just that heterogeneity exists, but this work gives us a real-time mechanistic view of how it's generated and amplified, right down to the level of sister cells and how they duplicate their DNA.
Precisely. It moves heterogeneity from being just a static description after the fact to a dynamic process you can actually measure. They phrase it as having causal resolution at the scale where selection acts, the cell family.
Which helps explain how even temporary DNA damage can have these lasting effects, driving plasticity and potentially early tumor evolution.
Exactly. It fuels the engine.
Okay, let's crystallize this. What are the, say, 3 main takeaways for you listening?
All right. First, replication stress leaves behind these heritable DNA lesions, marked by 53BP1, and the daughter cell has to confront them immediately in its G1 phase.
Got it. Inherited damage is an early crisis.
Second, even temporary stress creates significant asymmetry between sister cells. They diverge instantly in damage levels and their cell cycle behavior. They are not born equal after stress.
Unequal fates from the start. And third,
the route to polyploidy, that whole genome duplication, really matters. Re-replication is way more damaging to the genome than endoreplication. The path taken dictates the genomic cost.
super clear. It paints a much more dynamic picture of early cancer evolution.
It really does. And it leaves you with a pretty provocative thought, doesn't it?
Well,
if the fundamental difference between two potentially problematic sister cells is sealed right at that first division, because of how damage, or maybe just one protein like 53BP1, is inherited asymmetrically, could we potentially intervene right there at that very first unequal division before that divergence really gets amplified? Could that be a way to stop a tumor from developing its really aggressive, heterogeneous nature later on?
A very early intervention point, targeting that initial asymmetry. Fascinating stuff to think about.
Yeah, it opens up new ways to think about prevention or very early treatment.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.